Today, we're diving into a story of espionage, infiltration, and genetic hacking. And believe it or not, it happens every single day, all around us. It's basically a microscopic spy thriller, and our master agent is a bacterium, with its unsuspecting target being, well, a plant. So let's just think about that for a second. What if a bacteria could literally hack a plant? Not just infect it, but get in there and rewrite its fundamental DNA, all for its own benefit. It sounds like something straight out of science fiction, right? Well, it's totally real, and one little organism has been perfecting it for millions of years. So let me introduce you to our master agent, Agrobacterium tumefaciens. Its entire mission, its whole purpose in life, is to find a plant, breach all of its defenses, and then inject a piece of its own genetic code right into the plant's genome. And why? To turn the plant cell into its own private food factory. It's kind of genius and honestly a little bit evil. All right, so how on earth does this tiny little spy actually pull this off? Let's get into the mission briefing, starting with the key players and the secret code it's carrying. You know, every good spy has some kind of payload they need to deliver. For our little bacterium, that payload is a piece of its DNA called tDNA, and you can really think of it like, well, like a computer virus. Once this code gets inside the plant's main computer, it's going to completely take over and reprogram everything. Okay, mission phase 1. You've got to breach the outer defenses. For Agrobacterium, that's the plant's super tough cell wall and its membrane. It's no easy task. So first, a whole bunch of these bacteria swarm the plant's surface, kind of like setting up a base camp. And then they build this incredible little device, a tiny hollow needle called a T-pilus. It's basically a microscopic syringe they use to punch right through the plant's armor and inject that tDNA payload straight inside into the cytoplasm. But, you know, getting inside is just the beginning. Now the payload is in hostile territory. It's got to navigate the dangerous cytoplasm to get to the real target, the nucleus. That's the command center, where all the plant's genetic blueprints are kept. And this is where it gets super cool. The tDNA, our payload, it doesn't just wander around hoping for the best. Nope, it's got a pilot. A bacterial protein called VIR-D2 latches onto the front and basically steers the whole thing. This package deal is what we call the T-complex. Now, the cytoplasm is not a friendly place for foreign DNA. The plant's immune system is on high alert, ready to shred anything that looks suspicious. So what does Agrobacterium do? It sends in the bodyguards. Another protein, VRI2, rushes in and wraps itself all around the tDNA, creating this perfect disguise. It's like a stealth cloak, making the payload invisible to the plant's security. Okay, but this is where the plan goes from clever to absolutely brilliant. The spy package needs a ride, right? It can't just float around, so it finds an inside man. It actually hijacks one of the plant's own proteins, a guy called VIP1, and turns it into an unwitting accomplice who doesn't even know he's been compromised. So check this out. That VIR2 bodyguard protein, the disguise, it's designed to grab onto the plant's VIP1 protein. Once it's latched on, VIP1 is completely fooled. It thinks it's just doing its job, and it starts escorting this entire hostile package along the cell's internal highway system, the cytoskeleton, moving it straight for the nucleus. The ultimate betrayal. All right, we're at the final boss level, the nuclear vault. This thing is locked down tight, with guards at the gate, known as the nuclear pore complex. Getting inside is the whole ballgame. So how do they crack it? You guessed it, the inside man. See, our unwitting accomplice, VIP1, has a built-in keycard. It's got the security clearance to get into the nucleus. So by just holding on tight to VIP1, the entire spy complex basically flashes a stolen ID badge at the guards and waltzes right in. It's incredible. And here's the absolute kicker, the most brilliant part of the whole plan. VIP1. Its normal job is to be part of the plant's defense system. It's supposed to be the guy who runs to the nucleus to sound the alarm when there's an invader. But Agrobacterium gets to it first and uses the plant's own alarm system as a Trojan horse. Just wow. Okay, they're in. The package is inside the vault. So what now? Well, they don't need the getaway car or the disguising anymore, so a new bacterial operative, VRF, shows up. Its job is to get rid of the evidence. It tags VIP1 for destruction, the whole disguise falls apart, and the tDNA payload is released, right next to the plant's most sensitive data, its chromosomes. This is it, the final move, the point of no return. The tDNA has to go from being an intruder to becoming a permanent part of the system. It has to go deep, deep undercover by weaving itself right into the plant's own genetic code. And for its final trick, the bacterium does pretty much nothing. Seriously, it doesn't bring its own tools for this part. In one last amazing act of manipulation, it tricks the plant into doing all the heavy lifting itself by exploiting the plant's own DNA repair crew. So there are a couple of ways this can go down. The most common one is this quick and dirty method called non-homologous end joining. You can just call it NHEJ. It's basically the plant's emergency DNA repair system. The tDNA just shows up looking like a broken piece of DNA, and the cell's repair crew rushes in like, oh no, we gotta fix this, and accidentally stitches the enemy code right into its own genome. It works like this. First, the plant's repair proteins grab onto the ends of the tDNA, thinking they're holding a broken chromosome together. Then they act as a scaffold, calling for backup and bringing in all the other repair tools. And finally, the plant's own DNA ligase, think of it as molecular superglue, comes in and permanently stitches the tDNA into the chromosome. The plant has just repaired itself by installing the malware. Mission accomplished. So that's it. Mission success. The agent is in, the command center is compromised, and the target's programming is permanently changed. So, what happens now? What's the impact of this incredible microscopic heist? Well, in the wild, that tDNA code forces the plant to grow these tumors called crown galls. And those tumors pump out a special food that only agrobacterium can eat. But here's where it gets mind-blowing for us. Scientists figured this whole process out. They realized they could take out the bacteria's payload and swap in their own, genes for things like pest resistance or making a crop grow better in a drought. This natural little hacker has become the #1 tool for modern genetic engineering in plants. And that kind of leaves us with a really fascinating question to chew on, doesn't it? This whole process, this natural hacking, has been going on for literally millions of years. So it really makes you wonder, what does genetically modified even mean? Where exactly is the line between what nature does all on its own and what we do in a lab?